Okay, here we go on chapter 20, which is on antimicrobial drugs. And we're going to talk mostly about antibiotics. However, we're also going to talk about anti-fungi, anti-helminth, kind of all the classes. So let's get started. So once again, remember earlier on in the semester, I asked you to broaden your definition of what chemotherapy is, because we tend to think of chemotherapy as cancer treatment. However, therapy just means treating a disease. Chemo just means chemical. So it's really treating anything, and aspirin is technically chemotherapy for a headache. And in this case, we're looking at drugs that are going to, either inhibit or kill or destroy microbes. The corgis are fighting over a rawhide right now. That's what that growling is. Just, I'm not running a dog fighting ring. So the goal of antimicrobial chemotherapy is to administer a drug to an infected person, and you want to get rid of the microbe without harming the host. And we'll just assume for the scope of this chapter that we're talking about a human host. Okay, so microbial diseases that are occurring in humans. It's really hard to achieve this goal because, as I mentioned in the previous chapter, it's why a lot of drugs are given in microbistatic concentrations, because there's no such thing as a side effect free drug. So yeah, this is an awesome goal, but do we achieve it? We started to, and I'll point out that example. So we'll talk about where these drugs come from, as to their range, what types of organisms, where did they come from? Are they from nature, or were they made in the lab, or are they kind of in between? So when you're considering selecting an antimicrobial drug, there's three things that you have to know. Okay, first of all, you need to know what is causing the infection. Okay, so if it's bacterial, then you can select an antibiotic. If it's viral, you wouldn't prescribe an antibiotic because it's not going to work. You would consider perhaps an antiviral. If it's fungal, then antivirals and antibiotics aren't going to work. So first of all, you have to figure out what's causing the infection. And a lot of times in healthcare, because you guys don't have a rapid test for everything, it's really a hypothesis or your best informed guess. Then you want to know, is that microorganism susceptible to various drugs? Are drugs even an option? And then also, and this is where it becomes really important to take a history from your patient, you need to know about the overall medical condition. So do they have allergies to medications? Do they have high blood pressure, diabetes? Are they, unwell to begin with, and maybe this drug might be too toxic for them? So there's, a lot of things to know. So as I mentioned here, we would love to have a rapid test. We don't. And those rapid tests also, like any test, are not completely foolproof. And in fact, some of them have high incidences, or higher than we'd like to see, of false negatives, meaning that the person is actually positive, but perhaps they don't have enough of the organism showing up to cross the threshold as being found positive by the rapid test. If you were to test them again in a week or a few days, then they might be positive. But that's one of the problems that we see with rapid tests. So it's going to be your experience. You're going to be a lot better in 10 years, knowing what you think someone has when they walk in the door to your treatment room, as opposed to just starting out in your career. But it really it's a hypothesis. You guys aren't, superhuman. You don't have a crystal ball that tells you exactly what's going on in that patient's physiology. So yeah, sometimes you try one thing and that drug is ineffective, and then the patient comes back and they're still sick, and then you have to reevaluate the data and come up with another hypothesis. Patients get mad about that. They expect you to be superhuman and to fix everything the first time, but guess what? That's not reality. Okay, and then also there's testing done, thankfully, in pharmaceutical companies for how susceptible these microorganisms are to particular drugs. So I know this looks like a big list, but this is kind of like the list from the in chapter 7 where we talked about, the ideal disinfectant or antiseptic. This is our dream list. Are we going to hit everything on the list? No, and a lot of it is common sense. Okay, so the biggest thing is this. We want that drug to go in and destroy the microbe, but leave our cells alone. We would love it if it was microbicidal, or in the case of viruses, just was able to obliterate, and inactivate all of those viruses. Because of the toxicity of drugs, normally these drugs function in microbistatic concentrations. Okay, we want it to be soluble. So, if you swallow a pill and it's not absorbed, the drug isn't absorbed into the bloodstream and you just poop it out the other end, doesn't do you any good. Okay, we want it to be able to function when it's highly diluted in body fluids, and we need it to remain in your system long enough to do what it's supposed to do. If it's excreted, metabolized and excreted in a few hours, that's not doing you any good. We don't want it to lead to the development of antimicrobial resistance, or what we call superbugs. This a lot of time has to do with patient behavior. So I'll mention that in a little bit too. Obviously, we don't want it to destroy elements of your immune system because we need that to work with the drug. Like I said, we want it to be active. We need it to be able to be delivered to the site of the infection. And sometimes that can be tricky. So it's why, for example, there are very few drugs that can treat meningitis, because very few drugs can cross the blood-brain barrier. And so that's a hard site to get to when you're trying to get medications over to treat that type of infection. Obviously doesn't do us much good if it's, $10,000 a pill. And we don't want it to cause allergies or to predispose the host to other infections. A lot of times we can't control, and it actually does do that, unfortunately. Talk about that a little later too. So let's talk about antibiotics. Okay, so antibiotics, first of all, mankind can take zero credit for the development of antibiotics. Antibiotics were simply metabolic products of either, a lot of fungi. So, Penicillium, Cephalosporium, those are the genus, and then, which produce penicillin and cephalosporin. And then we also have some species of bacteria that produce them. And let me go back to our original slide here. So if you remember when I talked about Alexander Fleming, God, I tried to pinch my computer screen. I'm an idiot. I'm a boomer. So this is what Alexander Fleming saw. He was working with Staph aureus, and so the big smears there are Staph aureus. And he was working in New York City during the summer, no air conditioning at the time, so all the windows were open. And he noticed on one of his plates that it had been contaminated with a fungus. Now, this is why I would never win the Nobel Prize, because I would look at that and say, ooh, my plate's contaminated, and chuck it in the trash. Alexander Fleming, being the brilliant guy that he was, noticed this clear zone in between the fungi and the bacteria, and he surmised correctly that this was likely something being produced by the fungi that was either inhibiting or killing the bacteria. Lo and behold, he was correct, and he isolated penicillin from that. Okay, so purest antibiotics come from nature. However, scientists can look at the chemical structures of those, try to synthesize drugs that have an equivalent function. Those would be synthetic, and then, kind of in between would be semi-synthetics. The problem is that, we have overused antibiotics, and the problem is that now we have all these antibiotic resistant superbugs. And so now scientists are looking in, other species of bacteria and fungi, but also algae and plants, hoping that we find some new and novel antibiotics, and now we've learned not to overprescribe them. So the definition of an antibiotic is it is a chemical that is produced by one microorganism that can inhibit or kill another microorganism. So when you hear antibiotics, antibiotics come from nature, not from a lab. When you hear the term antimicrobials, those are usually drugs that come from a lab or lab created. When we talk about antibiotics, there are two main definitions. So a broad spectrum antibiotic is effective against all of the regular bacteria, okay, that can be gram stained. So obviously I'm not including mycoplasmas and mycobacterium in this definition, but broad spectrum antibiotics are going to wipe out both gram positive and gram negative organisms. Narrow spectrum antibiotics are effective against either gram positive or gram negative organisms. So ideally, when you see a patient, and let's say you've determined, all of the clinical signs point towards a bacterial infection, it would be awesome if you knew exactly whether the organism causing the infection is gram positive or gram negative. And then you would prescribe an antibiotic that just killed gram positives, because that's going to limit the, general destruction to normal flora in the patient. Ideally, the way to do that is to, take a sample and then, let it grow on agar, have someone in the lab do a gram stain, and then you get the results back. But a lot of times a broad spectrum antibiotic is prescribed simply because the patient is sick and you want to get on top of this infection and get them taking an antibiotic as soon as possible so the infection doesn't get any worse. And so that's why a lot of times broad spectrum antibiotics are prescribed first, and then if lab results come back, then you can always switch them over to a narrow spectrum antibiotic. But broad spectrum are great, but remember, they're going to wipe out the bad guys and the good guys, and especially the flora in your gut, and that comes with its own set of problems. So the goal of antimicrobial drugs, we want to, just mess up what's going on in those microbial cells. We want to mess up the bacterial cell wall. We want to get in and inhibit bacterial ribosomes. We want to do other things, to fungi and protozoans. Now, the problem with fungi and protozoans and worms, keep this in mind, these are eukaryotic, meaning that they are just like your cells. So you can't attack the DNA and the ribosomes of fungi, because you're going to be destroying your own DNA and your own ribosomes. So fungi, protozoans, helminths, anything that's eukaryotic, that presents a special challenge. And then, of course, the other class, you can't kill a virus, so you want to try to inhibit viral replication. So most of these drugs interfere with microbial enzymes that are required to synthesize or assemble things needed for the microbe to be able to continue its growth and physiology. So if you can shut those down, eventually the cell's going to die. One term that I definitely want you to know is here: selectively toxic, or another way to say that is selective toxicity. What that means is that the drug is going to either kill, inhibit, or inactivate microbial cells or viruses without damaging your tissues. Okay, that's the dream. Is it possible? Not 100%. There are some drugs that are better for that. Okay, so I'm at about 15 minutes. I'm going to stop here. We'll pick up with here in the next lecture. Thank you.